Hello, and welcome to Professor Gadgil GPAT Academy. Today we will discuss in detail about viruses. Viruses are infectious agents so small that they can only be seen at magnifications provided by the electron microscope. They are 10 to 100 times smaller than most bacteria. Viruses are incapable of independent growth in artificial media. They can grow only in animal or plant cells or in microorganisms. Thus, viruses are referred to as obligate intracellular parasites. Viruses largely lack metabolic machinery of their own to generate energy or synthesize protein. They depend on the host cells to carry out these vital functions. They have either DNA or RNA, but never both. The structurally complete, mature, and infectious virus is called as the virion. Viruses never multiply by binary fission and are unaffected by antibiotics. Now we will discuss the types or classes of viruses. Viruses multiply only in the cells of particular species, and thus they are divided into three main classes. First is the bacterial viruses or the phagenae. They have DNA and are called bacteriophages or simply phages. Second is the plant viruses, which is called as the phytophagenae. They have RNA and infect potato, sugarcane, tobacco, cucurbites, and any other higher plant. Third is the animal viruses, also called as the zoophaginae. They usually have DNA but may also have RNA and infect man, pigeon, parrot, dog, cow, arthropod, etc. Now we will discuss the classification of viruses. When not much was known about the structure or chemical properties of viruses, they were classified by where they were found. Thus, they have been classified as bacterial, plant, and animal viruses. They have also been classified as dermotropic, that is skin tissue, neurotropic, that is nerve tissue, viscerotropic, that is digestive tract, and pneumotropic, that is respiratory system, depending upon organs that they infect. Some scientists have even made the mode of transmission of virus from one host to another host as the basic character and classified viruses as enteric, respiratory, and arboviruses. As more and more knowledge has been gained about the structure of viruses at a chemical level, a classification has been developed based on the physicochemical properties. The viruses have been divided into families, which are split into genera and finally species. Viruses may be classified according to their nucleic acids. A nucleic acid that encodes information for making proteins needed by a virus is called as a positive sense nucleic acid. A nucleic acid made up of base complementary to those of a positive sense nucleic acid is called as a negative sense nucleic acid. The nomenclature of viruses, like their classification, is decided by the International Committee on Taxonomy of Viruses. Viruses are grouped into families which are named with the suffix viridae. Subfamilies, if any, are end with suffix virinae, and the genera have the suffix virus. Now we will discuss the morphology of viruses. A virion is a complete, fully developed viral particle composed of nucleic acid. Viruses are not cellular and therefore do not have a nucleus, cytoplasm, or cell membrane. Virus contains a single kind of nucleic acid, either DNA or RNA, which is the genetic material. The percentage of nucleic acid in relation to protein is about 1% for the influenza virus and about 50% for certain bacteriophages. The nucleic acid of a virus can be single-stranded or double-stranded. Depending on the viruses, the nucleic acid can be linear or circular. In some viruses, like influenza, the nucleic acid is in separate segments. The nucleic acid of viruses is surrounded by a protein coat called the capsid. Each capsid is composed of protein subunits called the capsomers. Capsid also serves as a vehicle of transmission from one host to another. Some animal viruses are released from the host cell and coats the virus with a layer of the host cell plasma membrane. That layer becomes the viral envelope. Depending on the virus, envelopes may or may not be covered by spikes, which are carbohydrate protein complexes that project from the surface of the envelope. Some viruses attach to the host cells by means of spikes. Viruses whose capsid are not covered by an envelope are known as naked viruses or non-envelope viruses. Now we will discuss the classification of viruses based on the capsid structure. First is the helical viruses. Helical viruses resemble long rods that may be rigid or flexible. The viral nucleic acid is found within a hollow cylindrical capsid which has a helical structure. The genetic material is generally single-stranded RNA and in some cases, single-stranded DNA bound into the protein helix, example rabies virus, tobacco mosaic virus. Second is the polyhedral or icosahedral virus. Many bacterial, plant, and animal viruses are many-sided or polyhedral. The capsid of most polyhedral viruses have the shape of an icosahedron, a regular polyhedron with 20 triangular faces and 12 corners. Example adenovirus and poliovirus. Third is the envelope virus. They are roughly spherical but highly pleomorphic, that is variable in shape and in nature. When helical or polyhedral viruses are enclosed by envelopes, they are called envelope helical, example influenza virus, and envelope polyhedral viruses, example herpes simplex virus. Next is the complex virus. Some viruses, particularly bacterial viruses, have very complicated structures. These are called as complex viruses. Certain bacterial viruses, that is bacteriophages, that infect E. coli, are called the T-even phages, that is T2, T4, T6. They are tadpole-shaped with a hexagonal head and a cylindrical tail. The head consists of a tightly packed core of nucleic acid, that is double-stranded DNA, which is surrounded by a protein coat or a capsid. The size of the head varies in different phases, from 28 nanometer to 100 nanometer. The tail is composed of a hollow core or a contractile sheet surrounding the core and a terminal base plate which has attached to its prongs, tail fibers, or both. The base of the tail, six pins, and six tail fibers are attached to that bind to specific receptor sites on the bacterial surfaces. Phages T1, T5, and lambda of E. coli do not possess a contractile sheath, and the tails of the phages T3 and T7 of E. coli and P22 of Salmonella are short and non-contractile. Now we will discuss the life cycle of bacteriophages. Bacteriophages exhibit two different types of life cycles. First is the lytic or the virulent cycle. In virulent cycle, there is intracellular multiplication of phages followed by lysis and release of progeny virions. This is called as the lytic cycle. Second is the temperate or avirulent or lysogenic cycle. In lysogenic cycle, the phage DNA becomes integrated with the bacterial genome, replicating synchronously without any cell lysis. Now we will discuss the lytic cycle or multiplication of bacteriophages. The replication of virulent phages was initially studied using the T-even numbered, that is T2, T4, and T6 phages of E. coli. There are total 5 steps, which are as follows. First is the attachment or adsorption. Phage particles come into contact with bacterial cells by random collision. A phage attaches to the surface of a susceptible bacterium by its tail. Host specificity of phages is determined at the level of adsorption. The infection of a bacterium by the naked phage nucleic acid is known as the transfection. Second step is the penetration. The phage DNA is injected into the bacterial body through the hollow core. Penetration may be facilitated by the presence of the phage tail of lysosome. After penetration of DNA, the empty head and the tail of the phage remains outside the bacteria, which are called as the shell or ghost. If many phages are attached to the bacterial cell, multiple holes are produced on the cell with the consequent leakage of the cell contents. Bacterial lysis occurs without viral multiplication. This is called as lysis from without. Phages such as T1 and T5 that do not have the contractile sheath also inject their nucleic acid through the cell envelope by adhesion sites between the inner and the outer membranes. Next step is the biosynthesis of phage components. After infection and penetration of DNA, transcription of a part of the viral genome produces early mRNA molecules which are translated into a set of early proteins. These serve to switch off host cells' macromolecular synthesis, degrade the host DNA, and start to make the components of viral DNA. The viral DNA replicates and also starts to produce a batch of late mRNA molecules transcribed from genes which specify the proteins of the phage code. These late messages are translated into the subunits of the capsid structures, which condense to form phage heads, tails, and tail fibers. Next step is the maturation. The process of assembly of the phage from its components is called as the maturation. Last step is the release of progeny phage particles. The release of progeny phage particles takes place by a sudden explosion or bursting of the bacterial cell wall. Lysozyme synthesized within the cell causes the bacterial cell wall to break down. Each cycle of phase reproduction may require 20 to 60 minutes, and a single phase infection may produce 200 or more progeny. The sequence of events by the injection of the phage nucleic acid and release of newly synthesized virions is called as the replication cycle or the viral multiplication. The interval between the entry of the phage DNA into the bacterial cell and the appearance of the first infectious intracellular phage particle is known as the eclipse phase. It represents the time required for the synthesis of phage components and their assembly into mature phage particles. The interval between the infection of a bacterial cell and the first release of the infectious phase particles is known as the latent period. Immediately after the latent period, the number of phase particles released increases for a few minutes till the maximum number of daughter phages is reached. This period, during which the number of infectious phages released rises, is known as the rise period. Now we will discuss the temperate or avirulent or lysogenic cycle. In contrast to virulent phages, temperate phages do not cause lysis of the host cell. Following entry into the host cell, the temperate phase nucleic acid becomes integrated with the bacterial chromosome. The integrated phase nucleic acid is also known as the prophage. The prophage behaves like a segment of the host chromosome and replicates synchronously in the bacterial cell. This is called as the lysogeny, and a bacterium that carries a prophage within its genome is called as the lysogenic bacterium. The prophage confers certain new properties on the lysogenic bacterium. This is known as lysogenic conversion or the phase conversion. A lysogenic bacterium is resistant to reinfection by the same or related phages. This is known as superinfection immunity. Every time the host cell machinery replicates the bacterial chromosome, it also replicates the prophase DNA. The prophase remains latent until or prophase remains latent within the progeny cells. However, a rare spontaneous event or the action of UV light or certain chemicals can lead to the excision of the phase DNA and to initiation of the lytic cycle. This is known as spontaneous induction of the prophase. Now we will discuss the cultivation of viruses. Viruses are obligate intracellular parasites. Three methods are used for the cultivation of virus. First is the laboratory animals. It is the oldest method. Reed and colleagues in 1900 used human volunteers for their pioneering work on yellow fever. Landsteiner and Popper in 1909 used monkeys for the isolation of the poliovirus. The poliomyelitis virus, after intraspinal or intracerebral inoculation in monkeys, causes a typical paralytic disease. The use of white mice, pioneered by Thiele in 1903, extended the scope of animal inoculation. Infant mice are very susceptible to Coxsackie and arboviruses. Smallpox virus may be inoculated in the sacrificed skin or cornea of rabbits. The growth of the viruses in inoculated animals may be indicated by death, disease, or visible lesions. Animal inoculation is also used for the study of pathogenesis, immune response, and epidemiology. Next is the embryonated eggs. The embryonated hen's egg was first used for the cultivation of viruses by the Good Pasture in 1931, and the method was further developed by Burnett. Fertile chicken eggs, incubated for 5 to 12 days, can be inoculated through the shell aseptically. The opening may be sealed with paraffin wax and the egg incubated at 36 degrees Celsius for the time required for the growth of the viruses. Viruses may kill the chick embryo or produce specific evidence of viral activity, such as production of pox on the chorioallantoic membrane and hemagglutinating activity in the harvested amniotic and allantoic fluid. These effects help in the identification of viruses. Now we will discuss the inoculation site and the type of viruses. Allantoic cavity, the type of virus is influenza and paramoxyviruses. In amniotic sac, we can cultivate influenza. In yolk sac, we can cultivate some viruses, chlamydia and rickettsia. In chick embryo, we can cultivate yellow fever 17D strain, rabies flurry strain, and both vaccines. In chorioallantoic membrane, we can cultivate vaccinia and herpes virus. Next is the tissue culture. The first application of the tissue culture in virology was by Steinhardt and colleagues in 1930, who maintained vaccinia virus in fragments of rabbit cornea. Maitland in 1928 used chopped tissues in nutrient media for the cultivation of vaccinia virus. The most important method for the cultivation of viruses was demonstrated by Enders, Weller, and Robbins in 1949 for poliovirus. There are three types of tissue culture techniques. First is the organ culture. Organ cultures are useful for the isolation of some viruses which appear to be highly specialized parasites of certain organs. Example, tracheal ring organ culture is employed for the isolation of coronavirus. Second is the explant culture. Minced tissues may be grown as explant embedded in the plasma clots. This is not useful in virology. In the past, adenoid tissue explant cultures were used for adenovirus. Third is the cell culture. Tissues are dissociated into the component cells by the action of proteolytic enzymes such as trypsin and mechanical shaking. The cells are washed, counted, and suspended in a growth medium and distributed in petri plates, test tubes, or bottles. The cells adhere to the glass surface and grow out to form a monolayer sheet. Total three subtypes are present. First is the primary cell culture. They are useful for isolation and cultivation of viruses for vaccine production. Example, rhesus monkey kidney cell culture, human embryonic kidney cell culture, human amnion cell, chick embryo fibroblast culture, etc. Second is the diploid cell strains. Diploid cell strains are derived from primary cell or are derived from primary cell cultures, which is embryonic in origin. They are of a single type and can undergo 50 to 100 divisions before dying. They possess the normal diploid karyotype, that is set of chromosomes. They are useful for the isolation of fastidious pathogens and also the production of viral vaccines. Example, human embryogenic lung cell strains WI38, rhesus embryo cell strains HL8. Last is the continuous cell lines. They are a single type of cells, mainly derived from the cancerous cells. Standard cell lines derived from human cancers such as HeLa, HEP2, and KB cells have been used in different laboratories throughout the world. They are stored at minus 70 degrees Celsius. Example, human carcinoma of nasopharynx line KB, human epithelioma of larynx cell line HEP2, human carcinoma of cervix cell line HeLa, and baby hamster kidney cell line BAK21, etc. Detection of viral growth in the cultures. Virus growth in cell cultures can be detected by cytopathic effect, transformation, metabolic inhibition, interference, heme adsorption, and immunofluorescence. Now we will discuss the multiplication of human viruses. The steps of virus infection and replication can be divided into six sequential phases. First is the adsorption. In the case of influenza viruses, the heme agglutinin on the virus surface gets attached to the glycoprotein receptor sites on the surface of the respiratory epithelium. Destruction of the receptor sites by neuraminidase prevents the viral adsorption. Second is the penetration. There are four possible mechanisms which the viruses use to enter the host cell. Non-enveloped viruses enter the cell by endocytosis or engulfment by the invagination of the section of plasma membrane with the accumulation of virus particles in the cytoplasmic vesicles. This is known as viropexis. The possible ways are: translocation is the process by which the whole non-enveloped virus enters the host cell by moving across the cell membrane. Fusion is the endocytosis of enveloped viruses in which the envelopes of the virus fuse with the membrane of the endosome. Next is the direct fusion of the virion envelope with the surface membrane of the cell, which also takes place in some of the families of viruses. Next step is the uncoating. This is a process by which the virus loses its outer layer and the capsid. In some cases, uncoating is effected by the lysosomal enzyme of the host cell. In the poxvirus, uncoating is a two-step process. Next is the biosynthesis. DNA viruses synthesize their components in the host cell or host cell nucleus, except the pox virus, which synthesize their components in the cytoplasm. Likewise, RNA viruses synthesize their components in the cytoplasm of the host cell, except the orthomoxyviruses and paramoxyvirus and the leucoviruses. The critical step in viral biosynthesis is the transcription of mRNA from the viral nucleic acid. Transcription of messenger RNA from the viral nucleic acid. Second is the translation of messenger RNA into the early proteins. They initiate and maintain the synthesis of virus components and shut down the host protein and nucleic acid synthesis. Next is the replication of viral nucleic acid. And last is the synthesis of late proteins, which are components of daughter virion capsids. In reoviruses, the double-stranded RNA is transcribed into mRNA by the viral polymerases. Depending on the method of mRNA transcription, single-stranded RNA viruses are classified into two categories. In positive-strand RNA viruses, the viral RNA itself attaches as the M or itself acts as the mRNA. The negative-strand RNA viruses, that is paramoxiviridae, possesses their own RNA polymerases for the mRNA transcription. Last step is the maturation. Maturation may take place in the nucleus or the cytoplasm. Envelope viruses get enveloped from the cell membrane of the host during the process of budding. Non-envelope viruses are present intracellularly as fully developed virion. Last step is the release. The time taken for a single cycle of replication is about 15 to 30 hours for animal viruses. In animal viruses, release occurs without cell lysis as opposite to human viruses. Next is the cellular entry mechanisms. For non-envelope viruses, viropexis or translocation is present, and for envelope viruses, endosomal fusion and direct fusion is present. In viropexis, the cell swallows the virus into the cytoplasmic vesicles or the phagosomes. In translocation, the whole virus slides directly across the intact cell membrane. In endosomal fusion, endocytosis occurs, then the viral envelope melts into the internal endosome membrane. And in direct fusion, the viral envelope melts directly into the surface plasma membrane upon the contact. In the DNA virus synthesize components inside the cell nucleus. RNA exceptions are orthomoxiviruses, paramoxiviruses, and leucoviruses, which are synthesized in the nucleus. RNA viruses synthesize the components in the host cytoplasm. The DNA exceptions are poxvirus, which synthesize entirely in the cytoplasm. RNA transcription, positive strand RNA, viral RNA acts directly as mRNA. Negative strand RNA requires viral RNA polymerases for mRNA transcription. And reoviruses, double-stranded RNA transcribed to mRNA by viral polymerases. So, this was the complete guide for viruses. Like this video if it helped, share with a friend preparing for GPAT, NIPER or university exams. Subscribe and hit the bell icon. Comment the number of steps in the lytic cycle. Download the GPAT Pro app, which has 10,000 plus MCQs, 200 plus video lectures, 3 modules completely free. Link in the description. Join our WhatsApp and Telegram channel for daily MCQs and explanation. Link is in the description. Next video, antiviral drugs. Stay tuned. Professor Gadgil signing off.